Okay, so now what we need to do is continue with this chapter on cardiovascular system and get to finishing the cardio part and talk about the vascular part. So where we left off last time is we talked about the different ways that we can assess the heart activity in terms of auscultation or listening to the heart sounds and what that comes from, as well as looking at the electrical activity of the heart and using an ECG to determine what's going on in the heart. And when we saw it last time, what we saw is that we have three different repeating waves. We have the P wave, we have the QRS wave complex, and we have the T wave. And these three waves show the electrical activity of the heart. We know that when the heart gets stimulated by the autorhythmic cells, right, that are located in the SA node and the AV node and places like that, are different contractile cells that are actually going to make the heart contract are going to get stimulated. They're going to depolarize. And so what we find is that at the P wave here, all of the muscle cells in the atria, which are the upper two chambers, are going to contract. So P wave represents atrial depolarization, and then they're going to contract. And then the atrial, what's called repolarization, where they are going to then relax, comes right after during the QRS wave complex. Now the atria contract first and then the ventricles. At the time when the atria are relaxing or repolarizing, the ventricles are actually contracting or getting stimulated or depolarized. So this QRS wave complex is atrial repolarization, but it's also ventricular depolarization. After ventricular depolarization, where we stimulate those muscle cells to contract, then the ventricles are going to repolarize and relax again. So it's this alternating cycle, right? Atria, then ventricles, atria, ventricles, atria, ventricles, atria, ventricles. The atria contract first and then relax, and the ventricles contract and then relax. So it's got to alternate that way, because if all the chambers of the heart contracted at the same time, the blood wouldn't be able to move through the heart. Okay, so what's going on in terms of the heart is actually, kind of twofold. It's got intrinsic, basically localized control in terms of the SA node, the sinoatrial node, and how it works to coordinate the heart's activity internally within the heart. But we also have what's called extrinsic control. And that means that outside of the heart we have some sort of control that comes from the nervous system. And in this case it comes from the autonomic nervous system. Now back when we were talking about nervous system, we said the autonomic branch is basically the same as what we can refer to as the automatic branch. It's subconscious control of different organs and tissues around the body. And so autonomic nervous system is going to tell the SA node what to do. So, the contractile cells of the heart are kind of like the workers of the heart. The SA node is kind of like the shift manager telling the workers what to do. But then, of course, there's always the main manager of the store or the owner of the store that tells the shift manager what to do. And the nervous system, specifically the autonomic nervous system, is like that owner of the store. So there's both sympathetic and parasympathetic impulses, right? Sympathetic is fight or flight, parasympathetic, rest and digest. So sympathetic impulses coming down to the SA node are going to speed it up, while parasympathetic are going to slow it down. Now where do these impulses come from? Well, these impulses, if you also remember back from nervous system, we said that as you go lower in the brain, we get to more and more basic kind of life-preserving functions. And so what we find is that the cardiovascular control center is located as far down the brain as you can go in the medulla oblongata. And so the medulla oblongata is what is going to regulate heart rate. Now with any homeostatic control mechanisms, we're going to have that negative feedback loop. And so what we're going to have is we're going to have some type of receptors that's going to be telling the control center what's going on in the body so that it can make changes as necessary. And so the receptor in this case oftentimes is going to be baroreceptors. Now a barometer, as you may know, is a meter that measures pressure, like to measure atmospheric pressure. If you can tell, like when there's a high pressure system or when rain's coming and stuff like that. And a baroreceptor also senses pressure. And so pressure, like blood pressure, is certainly an important aspect to be monitoring in terms of the cardiovascular system. Okay, now in terms of this system, we saw already that there are two parts to the system, right? There's the cardio part and there's the vascular part. So it's connected in a loop, or actually three loops, right? There's what's called a systemic loop, a pulmonary loop, and then a cardiac loop. But in any case, it's a loop nonetheless, and that means that anything that occurs along the loop will have an effect around other parts of the loop. So anything that occurs with the heart, in fact, will affect the arteries and the vessels, etc. And likewise, anything that happens with the blood vessels also will affect the heart. And so what we find is that the blood vessels are certainly an important part. Now they do the carrying function of transporting the blood out to the tissues and then back again to the heart. Now when we look at them, we've got 5 different types of vessels. We have arteries, we've got arterioles, we've got capillaries, sorry about that line there, capillaries, venules, and then veins. Now by definition, we start with the arteries because the arteries always lead away from the heart. So that's the rule. Arteries lead away from the heart. The A's match up. Arteries away. Now most people think, oh, arteries are going to be red. They're always red. That's not always the case. It is the case if you're talking about what's called the systemic circulation, but it's not the case if we're talking about the pulmonary circulation to and from the lungs. So the rule instead is actually that arteries always lead away from the heart. Therefore, veins always return blood to the heart. Now, in terms of our other blood vessels, the arterioles are basically just small arteries, and the venules are basically small veins. So they kind of behave with the same rules, either away or to the heart. The capillaries are a bit different though. The capillaries are very, very important. Those capillaries, rather than transport like we talked about in terms of arteries and veins and arterioles, etc., those blood vessels, the capillaries, specifically are the site of exchange. And that's a very big concept in terms of the cardiovascular system because, you know, we think about it in terms of like delivery or, you know, going out to the tissues, but remember there's exchange that occurs out of the tissues. It's kind of like your, you know, postal service. They come, they drop mail off, but then they also pick mail up. If they were only doing one aspect of it, like dropping mail off and not picking up, they'd only be doing half their job. And it's the same thing in terms of the cardiovascular system. So these capillaries are the site of exchange. So that means we're going to be dropping things off. That means we're going to be picking things up. Okay? And these are the smallest blood vessels in the body. They're the smallest and the thinnest, which allow them to actually be the easiest place for molecules and ions, etc., to exchange. Okay, so we'll start with the arteries and the arterioles. These ones are the closest to the heart. They're receiving the blood directly from the heart. So when the heart contracts and it sends a big tidal wave of blood out to the body, the first vessels to get it are going to be the arteries and then the arterioles. So with our larger vessels, all of these vessels are going to have 3 layers or tunics to them. They're going to have an interior one, which we call the tunica interna, like internal. They're going to have a middle one called the tunica media, and they're going to have an external one, an outside one that's called the tunica externa. When we look at them and we compare an artery, like you see on the left, with a vein, we can see that they have all 3 layers, right? There is the tunica interna, the tunica media, and the tunica externa. The interna is actually epithelial linings on the inside of the blood vessel. The media is actually smooth muscle, and the externa is connective tissue. Okay? Now, in an artery where it's receiving these big tidal waves of blood, it's going to be very important that the arteries have a relatively thick wall to be able to withstand these big changes in blood pressure and blood flow. And so what we find is that they have a relatively thick tunica media or smooth muscle layer here compared to a similarly sized vein, like we look across here. Okay? Necessarily then it has a smaller lumen or space on the inside also compared to a vein. So these arteries are strong blood vessels that can withstand the pressures of these pulses of blood that are coming down. And that also relates to this idea when you talk about taking a pulse, you always are going to be taking a pulse on an artery because it's only in the arteries where the blood actually pulses because it's coming straight from the heart there. And as the ventricles contract, because they're the ones sending the blood out to the vessels, as the ventricles contract, they will then send this big wave through. And then as the ventricles relax, then there's the low pressure that's occurring inside of that blood vessel. Okay, so arteries and arterioles, thick walls, thick tunica media, and a small lumen. Okay, capillaries. We said that capillaries are the main site of exchange. So necessarily, these are going to be small and they're going to be thin. Okay, they're a single layer of flattened cells, simple squamous epithelium. And so what we find is that these are going to be the site of exchange, like we said, but the amount of exchange or the amount of flow through these blood vessels is actually controlled by what are called precapillary sphincters. And the sphincter is a band of smooth muscle that, when it constricts, can cut off effectively that blood flow to that capillary bed. And so depending on what's going on in the body, you don't always need, blood going through all these different capillary beds. What you can do is, say for instance right now, your arm and your eye muscles might be going, but your leg muscles aren't going too much. And so we don't need a whole bunch of blood flow going through those as much as if we were like running or something else. So we can actually use these precapillary sphincters to control how much blood is actually going to those particular capillary beds. Okay. So we said that these are the site of exchange. That means we're going to deliver things to the tissues from these capillaries, and then we're also going to get things back from them. Now, what actually causes fluids and things like that to exchange with the tissues? Because we do certainly exchange fluids as well as oxygen and carbon dioxide and stuff like that. So what actually gets them to exchange? Well, for fluids, at the beginning, at the arterial end, right? This is the arterial end. And I'm actually drawing in red, so you can't see it quite as well. But at the arterial end, you have a lot of what's called hydrostatic pressure. The fact that there's always more blood showing up from the arterial to this capillary is going to give it a certain amount of pressure. And so when it does, that blood that's in there is either going to go straight through the capillary, so it can go straight through the capillary and then exit the venule, or the fluid that's in that blood as well as solutes can actually go out. And so it's like if you're exiting a concert or something like that. You exit a concert, there's a lot of people going out the exits. You have a choice. You can go with the flow, or you can actually, ditch out to the side. And so that's what the blood can do as well. And so what we find then is at the beginning of the capillary, we have the net outward movement because of this hydrostatic pressure. And that's good because we want to be delivering things to the tissues. But in fact then, when we get to the venule side of the capillary, we get a net movement back into the capillary. Again, that's what we want to do. We want to exchange. We want to give stuff out. We want to get stuff back. And that, getting stuff back, is actually due to a different force. That force is called osmotic pressure. Okay? Osmosis has to do with the movement of water. It's the diffusion of water. And it has to do with the concentration of water. But it also has to do with the concentration of those other solutes. And what we find is that comparatively, we've got a higher concentration of water here in the tissues and a lower concentration of water back here in this end of the capillary. And so we get a net movement of water and solutes back into the capillary at the end. Okay, so at the beginning of the capillary, we have movement out of the capillary to the tissues due to basically hydrostatic pressure. By the end of the capillary, we're now getting things back into the capillary due to osmotic pressure. Okay, so if we go full circle, we come to venules and veins. And venules and veins, like we said, are going to be returning blood back to the heart. Venules are just small veins. And these are going to be so far around this cardiovascular circle that we don't see any pulsing going on in them. And likewise, we didn't see any pulsing going on in the capillaries. The capillaries are so small and so delicate, excuse me, that in fact, they are only a single flattened cell layer in terms of their wall, and they're only about the size of a red blood cell. In fact, red blood cells have to generally go through these capillaries single file. They are so small. So we can't have any pulsing going on in the capillaries because they would basically blow up. So we stop the pulsing down at the arterioles and the arteries and arterioles. When we get to the capillaries then, very low pressure, slow flowing system, so we have a lot of time to exchange. Then we dump into the venules and back into the veins. Now, like we said, these are very far away from the heart, and so they don't have pulsing either. They don't need as much muscle in their tunica media as we saw in terms of the arteries. Now, the other specialized thing that they do have, though, are valves. So much like we have valves in the heart to make sure that we have one-way flow of blood through the heart, we also have valves inside of these veins. Okay? They also tend to serve as what we call blood reservoirs. And what that means is that because the blood is so slow moving through them, and because the wall can distend or stretch out, then they can hold a lot more blood than what we see in terms of things like the arteries. So they're commonly called a blood reservoir. Okay, a few other topics, physiological topics, before we finish up this system. Blood pressure. Now, we referred to blood pressure a bit already. We talked about how it's high pressure in the arteries near the heart. And in fact, when most people are talking about blood pressure, they're really talking about arterial blood pressure. And when we look at arterial blood pressure, we see, as we were describing earlier, two major times in arterial blood pressure and its kind of flow. We see these high pressure times during ventricular contraction when the blood is being squeezed out of the ventricles. And that is called systole. I know here it says systolic pressure, but you also see it just written S-Y-S-T-O-L-E, systole. And so systolic pressure has to do when the ventricles are contracting or the ventricles are doing systole. Okay? So that's when that big tidal wave of blood is coming down them. When the ventricles are relaxing then and no new blood is coming into the arteries, that's what's called diastolic pressure. So if you go to the grocery store or something like that and they have one of those little pressure cuffs there, you're trying to take your blood pressure, it's going to give you two numbers, right? It's going to give you a systolic pressure and it's going to give you a diastolic pressure. Systolic is always going to be higher than diastolic unless there's something really crazy going on in terms of your arteries and veins. Okay, things that can influence blood pressure. Having an exam, right? Yeah, that can do it. We'll talk about that. But basically, like we said, because this is a loop, things that happen in the blood vessels as well as things that happen in the heart can affect the overall circuit. And so arterial blood pressure depends on what's going on in the heart, depends on how much blood you have inside of the vessels, as well as what's called resistance, meaning that as the blood goes around, it kind of drags its way along the walls. And so if you have a very small blood vessel, like a arterial or capillary or something like that, there's a lot of resistance there. There's a lot of the blood that's actually dragging along the walls. And then also blood viscosity. So if something is viscous, that means that it's kind of like honey or oil. It's slow moving, it's very thick. So viscosity also has an effect on it. If your blood is like honey, it's not going to be very easy to actually push through these different vessels. Okay, control of blood pressure then. How can we control that to make sure it's maintained homeostatically? Well, there's two ways that we can do it. We can tweak what's going on in the heart, or we can tweak what's going on in terms of the blood vessels. What's going on in the heart is called cardiac output. Cardiac output is basically how many times a minute your heart is contracting, and then also how strongly or how forcefully it's contracting each time it beats. So that's basically cardiac output. When we talk about peripheral resistance, that has to do with the blood vessels. And so if we have, if we're constricting those blood vessels because we're stimulating that tunica media and making that blood vessel smaller, then what you do is you increase resistance at that point, and it gives you more resistance. Now, what's overseeing this is actually what's called our cardiovascular control center, like we mentioned previously, right? The medulla oblongata. And that controls both the heart, like we mentioned before, as well as the blood vessels. But there's a lot of other things, like we said, like exams or emotional upset, exercise, as you need to increase your delivery to and from the tissues. Those also can affect your blood pressure and blood flow. Now, on this kind of flip side of the cardiovascular system in the veins, we don't, like we said, it's pretty far away from the heart. It's low pressure and slow moving. And so one of the ways that we can, quote unquote, encourage, if you will, the blood to move around is a few different ways. Number one, we know that these blood vessels, these veins, actually have valves inside of them. And with the valve, just like in the heart, it's going to prevent blood from going back one way where it shouldn't go. Okay? So the veins have valves. They also depend on skeletal muscle contraction. And what that means is that as the veins are going through skeletal muscles, in fact, we find that when the muscle contracts, what it does is it squeezes the vein. It's like squeezing a toothpaste tube. Okay? You squeeze the toothpaste tube anywhere inside, what you get is toothpaste coming out one end and not the other end. Okay? Same thing in terms of the valve. So when you squeeze that valve with the skeletal muscle, what happens then is you actually get the blood going through the valve, and then it's not able to come back the other way. Okay? So it's always moving towards the heart. There's also the breathing movements. Because the heart exists within the thoracic cavity next to the lungs, any breathing movements you have actually overall creates a negative pressure in that thoracic cavity. And with a negative pressure in there, it works like a suction to draw blood back in. And then we have what's called vasoconstriction. Remember that while the smooth muscle in the veins is much thinner compared to arteries, there still is some there. And so necessarily then, when that contracts, you can actually get squeezing of that vein by itself, if you will, that is going to help to move that blood through again. And there's just that picture of the valve, the one-way valve, as the blood goes through, and then when it tries to come back again, it causes the valves to close and seal off. Okay, so you'll be happy to know that the very end of this chapter is actually diagrams, like we talked about last time. The very end of this chapter is going to be diagrams showing you different arteries and different veins, which is very beneficial for lab this week as you're doing arteries and veins. But in terms of actual lecture material, we don't need to spend much time at all in terms of the rest of the chapter. Now, if you look at it, I have them in here for reference, but it just shows how all the arteries are going all the way around the body to supply blood to all the different areas and tissues. And then also the veins that are going to drain all those different areas and work to bring it back to the heart. So you'll certainly use these, like I said, as a reference in terms of your lab, but not too much of a concern in terms of lecture. So for the most part, then, that ends our lecture on cardiovascular system.